see best in very dim light by Priya Mathur, Manasa Kalluri, and Abby Jackson. The eye. Before we did our experiment, we learned about the eye. We learned about the iris, the lens, the pupil, cornea, vitreous gel, optic nerve, macula, fovea, and retina. We focused on the retina. The retina. The retina is the back of the eye. Rods are a part of the retina that help you see the colors black and white. They also help you see things in dim or no light. Cones are the part of your retina that help you see the other colors. Use your cones during the day. For our experiment, we tested how well people could see colors in dim light. People used their rods to see the colors. The retina. These cone-shaped things are the cones, and these are the rods. This is a picture of rods and cones. These are the rods, and this is the cones, and these are the cell fibers. These are the three cone types. There are red, green, and blue. Red is 64%, green is 32%, and blue is only 2%. Blue is the most sensitive type of cone, but you don't have any in your central vision, so green is the most sensitive in your central vision. Experimental design. For our experiment, we wanted to test how well people could see different colors with small amounts of light that gradually increased. We used 15 color tiles: light and dark red, orange, yellow, green, blue, purple, and pink. We also used white. Before we had each person individually tell us which colors they could see at each light level, we had them sit in a dark room for 20 minutes so that the rod cells in the retina could be fully adapted to the darkness. Materials. To get the color tiles we needed, we went to Mott's paint store and took samples of light and dark colors. We also took white as a control. White was the control because it has all the colors in it. A large tray holds all 15 color tiles. Black felt put over the tray because the color of the tray may have affected the experiment's results. And we also created a table that we filled out with information during the experiment. This is a picture of the Mott's paint store. Visiting ophthalmology office. Before we figured out all the details of our experiment, we visited Dr. Michael Norrick's ophthalmology office. He showed us some of the equipment used to detect color blindness and gave us some ideas to use for our experiment. These two pictures are pictures of a colorblind tester. This side has the colors on it, and you arrange them from light to dark, and then the other side has numbers to check how well you did. These two pictures are pictures of Manasa and I looking through an ophthalmoscope. That's our mentor, Doris. This is Abby and Manasa in the car, and this is Abby and I looking at the issue book of numbers and colors. Hypothesis. I thought that orange would be the color that people saw best because it pops out from the rest of the colors. Manasa thought red would be the color people saw the best because it's bright, and Priya thought green would be the color that people saw the best because people's eyes are most sensitive to the color green. Procedure. First, we got people adapted to the darkness by keeping them in a dark room for 20 minutes. Then the person would come down and tell us what they thought each color tile was at light levels 1, 2, 3, 4, and 5, with 1 being the lowest and 5 being the highest. We put tapes on the dimmer switch to show each of the light levels. While they told us what colors they thought each tile was, we recorded the hour table. Data. The first set of collected data we took was during the experiment. This is what the table looked like. We wrote the subject, which is the person we tested on, the date, and the time that we tested that person. Data. After we collected all the data from everyone in that format, we converted the data into a numbering system. The number 1 is correct and the number 0 is wrong. The letters with numbers on the top are the number coding way to represent each color that we used. 1 is red, 2 is orange, 3 is yellow, 4 is green, 5 is blue, 6 is purple, 7 is pink, and 8 is white. The letter A stands for dark and the letter B stands for light. Then we converted the information from the other table to this. These numbers are the decimal value of people that got each tile right at each light level. These decimals were helpful because we were able to easily graph them. This is the red tile graph. Our y-axis was the frequency of correct responses, 0.2, 0.4, 0.6, 0.8, and 1, and the light level 1, 2, 3, 4, and 5, and we had light red and dark red. This is the orange tile graph. This is light orange. This is the yellow tile graph. This is dark yellow, and this is light yellow. This is the green tile graph, light green and dark green. This is the blue tile graph, that's light blue, dark blue. This is the purple tile graph. This is dark purple, and this is light purple. This is the pink tile graph, light pink and dark pink. And this is the white tile graph, which was the control tile, and we only used one shade of white, and it was called bright white. Conclusions. From our experiment, we found out that white was the color people were able to see the best. At all light levels, at least 70% of people were able to recognize white. Of the color tiles, not including white, the dark yellow was most easily recognized in dim light. 5 out of the 7 graphs showed us that people were able to identify the darker colors better than the lighter colors. None of us got our hypothesis right. Acknowledgements. We would like to thank the following people for helping us with our project this year. They all helped make our project successful. Doris Stobielzig, our mentor, Dr. Michael Nork, Colleen Jackson, Cam Jackson, Priya Mathur, Samir Mathur, Anjali Mathur, Sanjay Mathur, Sandhya Kalluri, Megan Kalluri, and Aisha Khan. This is our bibliography. We use Google, WebVision, Wikipedia, and more.